Hello everyone, my name is Juan Garcia, and I'll be presenting my work on hydrogel-mediated sequestration of cell-secreted granulocyte macrophage colony stimulation factor. So, during a bone injury or any injury, acute inflammation plays a key role in the regeneration process. But if left unchecked, it can lead to chronic inflammation. And during this chronic inflammation state, um, it can lead to a positive feedback loop of proinflammatory cytokines. Cytokines are just a class of proteins cells use to communicate to one another. And cytokines such as granulocyte macrophage colony stimulating factor, or GM-CSF for short, can inhibit osteoclastogenesis by suppression of CFOS, which is a subunit of the transcription factor activator protein 1, which essentially leads to in bone, decreased bone absorption and low levels of osteoclasts. So osteoclasts are a cell that can absorb bone, and it does this during a injury to essentially help remodel the bone in preparation for regeneration. So it's an important step. And our Our objective for this project was to sequester GM-CSF with an affinity-based biomaterial system. And how we approached this was first we needed to mimic the inflammatory environment. And we did this using human mesenchymal stromal cells, which are found throughout the body, not just in the bone. And we wanted to use a affinity-based hydrogel, and in this case, we were using a polyethylene glycol hydrogel system. And so how we go about this is using affinity proteins. Our lab uses an affinity protein called an Affibody. Affibodies are a small engineered protein that can bind to a lot of things. It consists of 3 alpha-helical structures. We were able to tune it to have a high, moderate, or low affinity to our target protein. And affinity just means how attracted our Affibody, in this case, would be to our target protein. So a high affinity means that our Affibody and our target protein will stay together longer, and a low affinity means that they're gonna only stay together for a short amount of time. And it contains 17 variable regions, which means that the Affibodies can have different amino acids in these regions that won't affect the structure, but will affect the overall affinity towards our target. So how we go about finding our Affibodies is using a yeast surface display library, which consists of 100 million Affibody variants, and we apply different cell sorting techniques such as magnetic activated cell sorting, which is essentially we have our target protein have a magnetic bead attached to it, so we can pull those Affibodies that have an affinity to our target protein away to identify, to essentially isolate, those Affibodies. And we also do fluorescence activated cell sorting, which essentially, instead of having a magnetic bead, we have a fluorophore, which essentially emits a signal to us, showing that it binds to an Affibody. So we then use a flow cytometry binding assay on our yeast surface display to quantify the dissociation constant, and that just tells us how long essentially our protein will stay on there. It is essentially like the affinity. And we just used varying amounts of GM-CSF to quantify that. And we then picked our high, medium, and low from those. And then we got the sequence from our yeast, put into bacteria, expressed our Affibody, purified it, and now once we have our purified Affibody, we then can make our hydrogel system. So our lab uses a 20 kilodalton 4-arm PEG maleimide polymer. So this polymer can react with our Affibody using the C-terminal cysteine on our Affibody, and it goes through a thiol-maleimide reaction, which results in the conjugation of our Affibody to the polymer. The remaining maleimide groups undergo cross-linking with dithiol-3-tol to create our Affibody-containing hydrogel. For this study, we are looking at different molar excesses of our Affibody in our hydrogel system, and because of that, we know it's going to be taking more of those maleimide groups up, so our hydrogel mesh is not going to be completely uniform, and there's probably going to be an increase in size. So we used equilibrium swelling theorem to investigate the mesh size. It takes into account the dry, relaxed, and swollen weight of our hydrogels, and what we saw was that with an increase of Affibody in the system, there was a slight increase, but it was not significant. We then used circular dichroism to confirm our Affibody structure. And circular dichroism is a technique that measures the differential absorption of left and right-handed circularly polarized light. And through this, we found that our Affibodies exhibited a negative band, two negative bands, one at 222 nanometers and another at 208 nanometers, and then also had a positive peak at 190 nanometers. We then compared it to literature, and when comparing, we found that it did coincide with a alpha-helical structure, which is what we predicted. So it was just further confirmation. To mimic the inflammatory environment, we grew our mesenchymal stromal cells to a confluency of 70 to 80%, and then we replated them and stimulated them with IL-1 beta for 24 hours. When stimulated, we collected the stimulated cell media and tested for the concentrations of GM-CSF within the media. And in this case, we grew for donor cells, and we found that the D140s produced the most GM-CSF, so those were the ones that we moved along with into the experiment. So then we incubated our hydrogel systems with the cell media, and after that we then analyzed how much GM-CSF was left after 24 hours of incubation, and we found that the 2000 molar excess Affibody-containing hydrogels sequestered the most GM-CSF. It sequestered about 70% of the GM-CSF in the media, which is a very good sign because we want to decrease the amount of GM-CSF. We then wanted to see how well our hydrogel system retained GM-CSF in it. So we removed the stimulated media and added fresh media, which contained no proteins. And after 24 hours, we quantified how much GM-CSF was left with a enzyme-linked immunoabsorbent assay, or ELISA for short. And we found that the 2000 molar excess hydrogel released the least amount of GM-CSF, releasing only about 20% of what it encapsulated, which is very good, because we don't want that GM-CSF going back into the already inflamed environment. So in conclusion, we were able to effectively decrease the concentration of GM-CSF with our affinity-based hydrogel system. And we found that increasing the amount of Affibody decreased the amount of GM-CSF released from our system. And the overall mesh size was not affected by the increase in Affibody in our hydrogels. It increased a tiny little bit, but it was not a significant amount. So I would like to thank the WUSI Human Performance Alliance for funding my summer research, the McNear Trio program for helping me throughout my research journey, and the NSF Career Award for paying me throughout my research through the academic year. And I would like to give a special thanks to the Hidarachi lab, which is the lab that I did all of this in. They're wonderful people, and I hope to continue my work with them. Thank you, and have a good day.